Thanks. So I will have to share my screen, right?
Yeah.
Okay. Oops. What are you seeing now?
You can see your background, like your screensaver, not your screensaver,
Okay.
like your home screen.
Now,
Yeah, that's good.
why are we social?
Yeah.
Yeah. Okay. So thank you everyone, first of all, for joining me. This is an opportunity to talk to you a bit about what I did at the Birkbeck Babylab, first as a PhD student and then as a postdoc. And really, the question that I write here, why are we social, is a huge one. And I am not going to answer to it completely with my talk, but I started little, just going and investigating whether we could find some genetic and brain signatures, really, that were associated with something important in social development. So how we interact with people and what do we prefer of the social world in which we are. And so why am I interested in that? Because when we look at infants and children, we see that they seem quite natural in socializing, really. And there are also studies that show us that since the beginning, since they're newborn, they are very much drawn towards configuration of the face. So when they see images with two eyes and a dot that suggests it's a mouth, they really prefer that stimulus, even if they haven't seen any face before. So this kind of suggests that there might be something in our DNA that really tells us you're going to be a social being. But then also, if we look at these pictures and our children, really, we see that they do receive a lot of training in social interactions as well, because their life is surrounded by social stimuli. We're not able to survive without other social beings around us in the first days and months of life. So really, it's also something that the environment is training us a lot. And so I want you to understand what is it contributing to how we become social beings. And this is particularly important, I think, because then we can understand also where we could intervene to facilitate the life for people who don't really feel quite at ease in our social world. For instance, individuals with autism spectrum disorder. And so what we can observe in our children is really like that they interact with each other, they look at each other, they share things, they imitate each other, they learn by being in a social environment. But really what I want to understand is what can we see in what is going on in their brain since they're very little, and also is there something in the genome, in the DNA, that tells us that we are going to become social beings, and how are we going to be in the social world. So with my PhD, what I did, this is me at the beginning of my PhD, collecting DNA from saliva samples from individuals, and I was gonna link what I could read in the DNA of people of infants with their brain activity that was recorded using electroencephalography. So it's a net. Some of you might be familiar with it because you've visited the Babylab before. But it's basically a net of channels or like little electrodes that record the electrical activity that is going on the surface of the cortex that is underlying our scalp. And it's totally a non-invasive technique. And actually, the baby you see here is my firstborn. He's done a lot of EEG sessions. So our goal was to link genetic information with the brain activity that we could see in children who were looking at faces on a screen, like social stimuli on a screen. And then we could link these as well with information about behavior coming from in-lab, in-person assessments, but also from questionnaires completed by parents, so that they could give us a hint of how the babies really behave in their social environment, like a home or daily life situations. And so I was lucky to be part of the BASIS team, so looking at data that belongs to the British Autism Study of Infant Siblings. And again, you might have heard about this study before. So we recruited infants when they were around 4 to 10 months of age. Some of the children had an older sibling with autism, so a family history of autism. And some of them had no family history of autism. And then at 3 years of age, the same infants, children, underwent a diagnostic assessment by a team of collaborators, clinical researchers at King's College in London. And so we could really understand who showed signs of a typical developmental trajectory among the infants with a family history of autism. So if they had a typical development at 3 years, some of them, it's usually the 20% of them, so a much higher proportion than what you would see in the typical population, where around 1% of the children develop autism. So around 20% of the infants with a family history of autism do develop autism by 3 years of age. Some of them do not quite meet criteria for an autism diagnosis, but they can show signs of other developmental difficulties. So sometimes in our research, we combine these groups together because we're interested in understanding what happens when you don't have autism. Sometimes we keep the groups separate depending on the research question. And then we compare these groups usually with a group of children with a typical development, so with no family history of autism. And so in between these times, we collected our EEG task around 8 months of age. And so I could look at the neural signatures that were meaningful for a later autism diagnosis. And with my PhD, I went to investigate whether there were genetic signs signatures that were associated with the brain, signatures that I could see at 8 months. And so this is probably the most technical slide of my talk, and I apologize, but I figured if people are joining this talk, they kind of are interested in the tools we're using. So I hope you can listen to this as well and make sense of what I'm saying. So what is DNA? DNA is really like a sequence of letters or bases, actually, that code for, ultimately code from proteins that serve to make us what we are. And so the majority of, so all individuals have the majority of the DNA that looks identical because we're all humans and we all do the same thing. So 99.9% of our DNA is really identical. But there are some locations that we have identified where there are differences between people, and those differences are totally normal and common, and we call them SNP. So it's these locations you can carry a different base from your neighbor or from anyone else in the world, and these differences make us really unique. So for instance, my height will depend on some of these SNPs, a combination of these SNPs, or my hair color, or also some psychological traits, and autism is among those. So for instance, individuals who carry A at this SNP location would look like the purple person. Individuals who would carry a G would look like the blue person, and individuals with a T would look like an orange person. So suppose that G is also associated with autism, and we can give it a weight. So say if you carry a G, you have +1 probability of getting autism. And this probability is so tiny that it doesn't matter really, but it is there in your genome. And then maybe also T is associated slightly with autism, so you would get a little bit increase in likelihood of developing autism if you carry a T in this location. And as you can see here, like orange individual could have a T there, but not the G at the previous location, so had a bit lower likelihood of developing autism. It might be that if you carry a G at that specific location, you are less likely than normal to develop autism. So in the end, if we sum up these weights for each individual, we end up with what we call a polygenic score, which is a fancy word to say it's a score that tells us how much is our likelihood of having autism, given previous studies that have identified which bases are associated with autism in the population. And so the majority of people would have a score of 0, but some of the people will have more of the autism-related genes. They're not really genes, they're locations in the genome, but some of them are located where genes are, some of them are not, but we call them genes to understand each other a bit more easily. So what we do is we compute a score. So purple individual would have a score of -0.5, so be placed there in the distribution. And then blue individual would have a higher score, and orange individual would have a +0.7. And so we get this individual score for each person. And so I computed that for the infants who were part of BASIS. And then I linked it with a signature of their infant brain response to social stimuli. So about the brain signature that I used, it's from EEG. And it's recorded while infants were looking at faces with a direct or averted gaze, or to a control stimulus that we call sometimes noise. And it's basically we took the pixels of the image face and we scrambled them so that you don't see the configuration of the face anymore, but you have a good control of, like you can see the same shape and the same colors in the image. And we look at the electrodes or the channels that record the posterior activity of the brain, because this has been linked with the development of face-related areas. And in adults, this is a pretty good marker associated with autism. And so our colleagues, Georgia Busu and Charlotte Tai, had shown that this signature is really predictive of autism at the individual level for the infants. And what we show here is that it is also mildly associated with this polygenic score. So if you carry more autism-related genes, you are more likely to be more engaged with the non-face stimulus, with the noise stimulus. And if you carry less autism-related genes, you have a longer brain activity or longer engagement with the face stimuli that we presented to the infants. So this kind of tells us that there is a genetic contribution, we would say, to what brain activity in response to social stimuli are as early as 8 months of age, around 8 months of age. And is this so we asked, is this meaningful and associated with behavior? And so again, I was linking this information that we had from the brain. In this study, I focused on the anterior parts of the brain, so these electrodes that you see colored here, because they we could compute the so-called ANSI component. So it's a signature that has been very robustly associated with attentional engagement with the visual stimulus. And so this is represented by a negative wave that is going on in the brain around 300 milliseconds after the infant has seen the stimulus. So if we look at the group of infants without a family history of autism, we see that at time 0, the stimulus is presented, and then we compute the average amplitude of the brain activity in these frontal areas, and we see a negative deflection in the purple area there, so around 300, starting around 300 milliseconds after the stimulus is presented. And we also computed the NC, so this wave for the other stimuli that we had, and we compared it between groups. And as you can see here, what the noise, the activity in response to the noise stimulus was much stronger, so more negative, for the infants with a family history of autism. And if you look at the solid red line, so family history infants with later autism show a much reduced negativity, so negative wave, in response to faces with direct gaze. And these are results as a group, but they tell us that really, if we go and look at this neural signature, we can see that already at 8 months, something is different for children who later develop autism. And so we also linked this with what parents reported us about the social behavior of these infants at home at 3 years. And it was again associated with this behavior. So it's really, we can see a link. It seems that we are social because we start to be engaged differently, to be more engaged with faces as early as 8 months of age. And then people with a different developmental trajectory can show less of this engagement. So it's really worth, we figured, looking at this a bit more at the individual level and understand whether this can predict how the individuals are going to develop socially later on. So we ask how, by looking at these brain signatures at the individual level, and this is what I'm doing with my postdoc. It's a project we called Behavior and Online Neuroimaging to study the development of socialization, so bonds. And we are using a method that has been developed by other scientists, and is called neuroadaptive optimization. Rianne Hartson and Pedro Costa, that you see on top here, are developing this using EEG with adults, and Elena Throm and I are in charge of the application for the infant study. So if you were with us earlier this year at a coffee talk, you might already know about this from Elena, because she presented the framework earlier on. So how does this work? We have a pool of stimuli that can be very different, and we want to understand which are the stimuli that elicit the strongest brain response in our infant. So first of all, we start with one image, And we present it to the infants. So here is, you see, we're using a slightly different cup than what you saw before. It's a wireless EEG system, very portable, that will be used in the toddler lab as well, what Tamsin was mentioning earlier. And so the infant wearing just this swimming cup with some electrodes on top would look at a screen with an image there, and we are recording EEG activity in real time. And then again, in real time, for that stimulus, we are computing these NC components, so the negative way, this neural signature that we were interested in, and that represents how much the brain is engaged with the stimulus that the child is seeing. And then we will feed this information to an algorithm, which is like a computer-based calculation that allows us to, again, look among our stimuli and try with another stimulus that the algorithm thinks could elicit a stronger brain response for the infant. And then we will test this again and run some iterations until we find really what is the stimulus that is preferred by that individual child. And so I have here an example of how it works. This is my baby and me at home, just to say that this tool is really portable. And so what you see here is, so I'm dragging his attention to the screen and presenting a face image, mum's face image. So he's really engaged with that as well. And then we present it a couple of times to get rid of noise, and then the algorithm, the real-time algorithm will calculate the NC component and choose another image that it wants to know the NC component for. And so now stimulus will be presented, and I can check the infant's engagement with the screen to make sure that I'm only recording activity when he's looking at the screen, so I can play that red thing to drag his attention to the screen again. And then a new NC will be calculated, and this will go on some more times until I see what is really the preferred image of my infant. And so we have had some infant scientists joining us in the lab before lockdown and in between the two lockdowns. And so we have learned something from them already. And that is that really the response to mom, the red one here, is stronger than to non-mom. And this is data from one individual child. So it's really powerful signal that we could obtain. And it's different from previous studies where we merge all the children together, and we don't really know whether this is true at the individual level. And so the answer, what have we learned, is that mom wins so far, and then we can use this information. We have validated the method, and we want to use this information to explore this a bit further. So we want to know, are there individual differences between infants and how similar the face of the mom should be to look like, to be engaging. So we can use these morphemes and present different types of stimuli to understand what level baby would prefer mom compared to Michelle Obama or someone else. We can also try whether infants prefer different combinations of gaze and facial expressions. So again, the infant could see mom looking at different directions and with different degrees of a smile. And we can see how their brain is responding to that. And it can really be that infants show differences in this. Because, for instance, there's evidence that infants with later autism are not engaged with the direct gaze as well as infants with atypical developments, that can be really informative at the individual level. And then Elena is also going to develop, as part of her PhD, a similar paradigm, but looking at live interaction. So if we can see what strategies the adult uses in interaction that elicit the strongest activation of the social brain in the infants. So this is really something that I also wanted to show, because we're working on, and it's I hope to do it like quickly. We're also interested in understanding whether infants are interested in voices in a particular level. And again, so far the results we have are only summarizing data at a group level. And as you see here, looking at these brains, the blobs show the activation in response to vocal sounds when compared to non-vocal sounds, and infants without a family history of autism show a stronger activation and in different areas compared with children with a family history of autism. And we can look at the slices in the brain, like images that represent slices, to see these differences here. And what we're doing now is that we take collected data from these typical likelihood infants, and we're looking at individual responses of each infant to each sound. So for instance, how would they reply to this type of sound, like human sounds, but without an emotional balance, or with non-voice stimuli. And these are individual, so it's quite novel. And so to sad sounds, all these sounds have different auditory properties as well. So we can really investigate what does the infant prefer, and is it linked to the auditory properties of this. And so this is a new study that we're running at the Birkbeck and UCL Center for Neuroimaging. It's called TAPS. And infants will go into an fMRI scan so that we can see the entire brain and not only the electrical activity in the cortex. And with headphones, we will play different sounds while they sleep. And we need them sleeping because this technique is very sensitive to movement. So they will be just naturally asleep while we play the sounds. And so for now, we're investigating what do parents think the preferred sounds would be for these infants, with an online study for 4 to 11-month-old babies. So if you have a baby scientist and you want to take part, you can be in touch with Elena. I'll just show you briefly how it looks like. This is me with long hair and with a baby. So you're asked to drag his attention to the middle, and then play a sound with your phone, and then rate how much, rate the behavior and how much you think the child likes the sound. And then you can try with it. You can try with other sounds. We have a mixture of social and non-social sounds, and we can see what parents think for this. There's another one, a very COVID-19 related sound. So please email Elena if at bonds_babylab if you want to take part, and she'll give you instructions. So to summarize, we found that we are social because our genes partly contribute, our brain get engaged with social stimuli, and now we really need to understand what does that individual infant brain prefer to understand if this is meaningful to understand developmental trajectories. And so we have these studies going on presented. So if you want to see what your baby prefers, just get in touch with us and follow us on Twitter and our website. I really want to thank my boss, Emily Jones, and Robert Leach, and Mark Johnson, but also all the bases and bonds participants who take part in the study, and you for listening. If anyone has questions, you can email me or ask now. I will just stop sharing my screen, I think, right?
Anna, thank you so much. That was absolutely fascinating and really, really clear explanations. I think you're the first person who's made genetics make sense. So well done.
Oh, okay. Yeah, I know it's a pretty sciencey, but I guess this is the purpose, like why people are joining our meetings.
Exactly. Does anyone have any questions? Please ask while we've got her here. Okay, well, I'll ask one. I loved how clear the response was to, well, I knew you were the mum in that case, but to the mum's face. Did you guys try using dad's face or like the nanny's face or whoever was the primary carer? Do you think that would have elicited the same response?
So thanks. It's a good question. So I must say that the one that the response that I showed was not even like my child's response, but was one of the bonds participants who came in, and they really all look like that a little bit. The responses are a bit sensitive to noise as well. So if children are tired, you get a less clear, really and deep response, but it's still quite consistent across babies. We haven't tried with other caregivers at the moment, just because we are still in the piloting phase and we really wanted to use the strongest signal we could get, and we based our hypothesis on the literature. So the literature only looked at mom so far. So that's why we chose mom. But this tool really gives us a lot of opportunities, and because of the real-time application, you can really finish the task quite quickly and try different things. And the baby is just wearing a swimming cap, so they're really fine with it for longer. We found that with very wiry hats, EG hats. So definitely something that we want to explore.
Excellent. And those of you with babies, hopefully the Babylab will be reopening next month. Certainly, I would have thought in April. So please, if this interests you or inspires you, please get in touch, because it'd be great to bring you back into the lab when the government restrictions relax a bit and it's safe to do so. And then you can see this in action. Does anyone else have a question? Oh, please, yeah, Fernanda. Oh, you're on mute.
Mute. Can you hear me?
Yeah.
Yeah, cool. Because she was saying about the baby's response to the mother's face. I guess it's because the mother was the person who was looking after that baby, was spending more time with that baby. So what if the baby had a carer who would stay with him for longer than the mother? Would that change the response of the baby? I don't know if she's gone that far down into the research.
Thank you so much for the question. It's really interesting. I think you're right. I think it would change the response. It's not based on my research, but it's about what people have studied about this neural signature before. It seems to be a measure of attention engagement, but it's also associated with familiarity. So I guess it makes sense that if a baby is more is cared by a person that is presented, whose image is presented on the screen, that person image would elicit the strongest response. That would be our hypothesis, But again, with this method, we could also see if there are individual differences in that. So if really what we're observing is how much the child is engaged with the face, we could really see there are differences between children, and it might be.
Okay,
You know,
thank
because the research is really based on group level responses. As a group, children behave like this, but it can be that individual infants are different.
Yes, thank you very much.
Thanks.